Hello dear students. In this session we are going to start a chapter biological classification. Okay, here we are going to discuss the introduction part of this chapter, and along with that in the end we will solve some questions to check what all we have learned. So let's begin. We will first discuss each and every line from NCERT because nowadays you know that in NEET questions are being asked from NCERT only, right? So we are here going to discuss each and every point in detail. First line of the chapter says that since the dawn of civilization, there have been many attempts to classify living organisms. Right, from the very old times, living organisms, they are being classified. Right, you all know that living organisms, there are so many living organisms. Okay, you have read about the chapter first, that is the living world. Okay, so living organisms, different types of living organisms are present on the planet. Okay, like plants, animals, bacteria, right? So all these living organisms, they need to be classified. But it was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, like for food, shelter, clothing, right? So in this paragraph, okay, till this line, what is mentioned over here uh, is that living organisms, right, they are being classified from the very early times, okay. But in the early times, okay, in the early times, the living organisms, they were classified based on, okay, based on the utility they provided to us. Okay, the utility, based on the utility they provided to us. Now, what is the meaning of this utility they provided to us? It means that there can be certain plants, okay, there can be certain animals, okay, which are edible, which can be taken as food sources, right? So such type of plants and animals, they were classified under this category, like food giving organisms, right? Some plants and animals, they were giving shelter. Okay, so they were classified in that category. Some they can give, some they can give clothing, right? So by this line you can understand that based on the need, okay, the need to use the living organisms, we used to classify them. And what you can find out here, that this is not scientific, okay, this is not scientific basis of classification. So classification system in the early times, in the previous times, they were non-scientific. They were based on non-scientific approach, okay, and they were based on the utility which they provided to us. Like some plants, they were edible, they were classified as food giving plants, food giving animals, okay. Certain large trees, they were giving shelter, so they were classified like that, okay. And certain plants and animals, you know that they can give fibers, they can produce fibers, right? So they were classified under the category of the use, that is clothing, okay. So now you understood that in the early times what happened, that living organisms, they were classified based on the utility they provided to us, okay. Now from here you can see there is a mention of this important scientist whose name is Aristotle, right? So first name you have to remember is of this scientist, okay. Aristotle was the one who tried to classify the organisms based on studying these scientific characters, okay. His classification system was based on the scientific characters, okay. Scientific approach he followed, okay. What scientific approach, what type of characters he chose? He chose, you can see, simple morphological characters, okay. Based on the external morphology of different organisms, they were classified on basis of that by Aristotle, Okay, so he chose scientific characters to classify organisms, and which characters? Morphological characters. What is the meaning of morphological characters? Meaning of morphological characters is what is the external features, you can say what are the external features, or you can say that which you can observe. Okay, you can say observable features, observable characters. Okay, like if you see a plant. Okay, like let's suppose you see a plant. Okay, and this plant is like a tree. Okay, so you will say this is tree. Okay, there is one plant which is very small. Okay, like this. Okay, so what you will say, this is looking like a herb. Okay, then there is one more plant which is very bushy and having so many branches, right? But it is not as thick, and you can say it's not like a tree. Then what you will say, it is a shrub, right? So this is how he classified plants. Okay, he classified plants under three categories. Okay, three categories based on what? Based on the morphological characters, or you can say features. Okay, and here you can see he divided all the plants based on the growth habit. Okay, based on the growth habit, he said that all the plants can be categorized as trees, herbs and shrubs. Okay, trees, herbs and shrubs. Okay, you can see here. Okay, you can see he used simple morphological characters, okay, to classify the plants into trees, shrubs and herbs. And he also divided the animals into two groups. Okay, one classification he gave for plants, and another classification he gave for animals. Okay, for animals, how he classified the animals? He said there are certain animals which are having red blood cells. Okay, animals with red blood cells, and some animals are without any red blood cells. So animals which are having red blood cells, he classified them, and one you can say name for them is there, that is called as enema. Okay, enema. And those animals which are devoid of RBCs, which do not have any red blood cells, they are called as anema. Okay, so these are the two classifications given by Aristotle, which you have to keep in mind. Okay, Aristotle, which you have to keep in mind. Okay, first classification system which was based on the morphological features. Okay, morphological features. And morphological feature is what? It is a scientific character. So from here actually the characterization or the classification of the living organisms just began. Okay, after this, who came? After this, very important scientist came, that is Linnaeus. Okay, Carolus Linnaeus, which you have learned in chapter Living World, who gave binomial nomenclature. The same scientist, what he did, he classified all the living organisms into two kingdoms. Okay, he classified all the living organisms, you can say all the living organisms, all the living organisms were classified under two groups. Okay, two kingdoms we can say. First kingdom was kingdom Plantae, and another kingdom was kingdom Animalia. Okay, what is this? This is called as the two kingdom classification. Okay, two kingdom classification. Who proposed two kingdom classification? Who proposed? Linnaeus. Okay, Linnaeus proposed two kingdom classification. You can see here. Okay, Linnaeus proposed this two kingdom classification. Okay, now what else he did? Which type of character he took into consideration? Right. So here you can see that he took only one character. Okay, which character he took? He divided all the living organisms based on presence or absence, presence or absence of cell wall. Okay, very important. Based on the presence or absence of cell wall, he categorized all the living organisms into two categories, Plantae and Animalia. Okay, so Plantae, in this group he kept all those living organisms where cell wall was present. Okay, where cell wall was present. And in Animalia, he kept all those organisms where cell wall was absent. Okay, now let's read what is written. Okay, in Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all the plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes. Okay, there was no distinction between eukaryotes and prokaryotes, unicellular and multicellular organisms, photosynthetic and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand. But a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. So, this classification system was discarded. Now, if we take very few characters, okay, first demerit based on only one single character. Okay. Now just think, if you want to classify something, then if you check and read and consider, or you can say observe many characters for that particular you can say thing, then it will be a more appropriate type of classification system, right? And if you will take only one character into consideration, then it will be an inappropriate type of classification system. Now just see, if we take cell wall, only this character, then how many different you can say organisms they can come into this category. Okay, I am writing here for you. Okay, just think, all the plants, you can say bryophyte, okay, algae, gymnosperms, pteridophytes, angiosperms, all they will fall into this category. Okay, then all the bacteria, okay, all the bacteria, they have cell wall. Archaebacteria, okay, cyanobacteria, okay, blue green algae, okay, cyanobacteria, okay, then what, fungi, okay, all the fungi, they will also fall into this category. Okay, some protists, okay, some protists, okay, especially the chrysophytes, okay, chrysophytes, dinoflagellates, okay, chrysophytes and dinoflagellates, they are having cell walls. Okay, so these members, all these members, they will fall into the category of kingdom Plantae. Okay, and others like all the vertebrates, okay, all the vertebrates, all the invertebrates, they are devoid of any cell walls. Okay, and mycoplasma, wall-less prokaryote, they will come under the category of kingdom Animalia. So just think that, is this classification system good? Can we use this classification system? No, right? How can we use this classification system? How can we consider this classification system? We cannot, right? Why so? Why I am saying this? Because here you can see that bacteria is a prokaryote. Archaebacteria prokaryote, cyanobacteria prokaryote, okay, right? All these groups are unicellular, okay. All these are multicellular. Fungi is multicellular, it is eukaryotic. Protist, they are eukaryotic and they are unicellular. So what is written here? That this system did not distinguish between eukaryotes and prokaryotes. Eukaryotes and prokaryotes are coming together. Single-celled and multicellular organisms, they are coming together like photosynthetic, and yes, very important, photosynthetic organisms and non-photosynthetic organisms, they are also classified together. So is this right classification system? All the plants starting from algae, bryophyte, pteridophyte, gymnosperm, angiosperm, and all these which are autotrophic, okay, which are autotrophic, can we keep them with fungi which is heterotrophic, right? So this is a wrong type of classification, right? Wrong type of classification system. And classification of organisms into plants and animals was easily done and easy to understand, but a large number of organisms did not fall into either category. Hence, this classification system was inadequate, right? So, This is a wrong type of classification system where you can see based on only one single character this classification system was. That is why it was incorrect. Okay, only one character which they took into consideration was cell wall, and who used this classification system? So Carolus Linnaeus. You have to remember all these things. Now, besides the gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Okay, classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. So here you can see that if we are choosing only one scientific character, that is the gross morphology, then also it is not a very promising type of classification system. As I have told you that a good classification system should have many characters. Okay, so for that other characters should also be included. For example, what is the structure of cell? How a cell is looking in an organism. In an organism, what is the nature of cell wall? Okay, cell wall is present or absent, and if it is present, then from which chemical that cell wall is made? Okay, what is the mode of nutrition, right? How that particular organisms it makes its food. Okay, it derives nourishment. What is the habitat? Okay, where that particular organism lives. Okay, and how the organism reproduces. And what is the phylogenetic relationships? How they are related with each other evolutionarily. Okay, so these are the different characters which we should consider. Okay, so many characters should be considered for a better classification system. Okay, classification system for living organisms have hence undergone several changes. So see, after the failure of two kingdom classification system, okay, after the failure of two kingdom classification system, there was a revision in the classification system many times. This was failed. Okay, then came three kingdom classification system. Okay, three kingdom classification. Okay, in this three kingdom classification, obviously as the name suggests, there were three kingdoms. Okay, kingdom Plantae, okay, kingdom Animalia, okay, and one more kingdom was made, that was kingdom Protista. Okay, and from kingdom Plantae and kingdom Animalia, some members were shifted to kingdom Protista, like all the prokaryotes, bacteria, okay, and you can say the dinoflagellates, okay, all those things, chrysophytes, they were moved over here. But then also this kingdom was having a confusion, right? In this kingdom, prokaryotes were present together with eukaryotes. Okay, same condition was there. Multicellular and unicellular were kept together. So it was inappropriate. So this was also failed. After that, one more classification system came, that was four kingdom. Okay, four kingdom classification system. Okay, in this four kingdom classification system, what have happened, that along with the Plantae and Animalia, along with the kingdom Plantae and Animalia, okay, then Protista, one more modification was made, that one more kingdom was formed, that was kingdom Monera. Okay, in this kingdom, from kingdom Protista, all the prokaryotes were kept. Okay, but then also there was a problem. Okay, then also there was a problem that after this, fungi, this was not getting a proper position, right? In this classification, three kingdom classification, prokaryotes and eukaryotes, multicellular, unicellular, heterotrophic, autotrophic, all were present together. So this was a problem. Here everything was fine, but fungi was not getting a proper position. For that, another classification system came into existence, that was five kingdom classification, right? We will talk about this, but now you have understood that why these three classification systems failed. So same thing has written over here, that several changes have been made over time, but kingdom Plantae and Animalia, they were kept constant under all different classification systems. You can see that I have made sure that there is no change there is no change in these two kingdoms. Okay, Plantae, Animalia, and here also only two kingdoms were there, Plantae and Animalia. So these were constant, but there was reshuffling of all the other members, like all the prokaryotic members. You can say the bacteria, cyanobacteria, protista group like euglenoids, okay, chrysophytes, all these were shuffled and shuffled again and again. Now, in this you can see same thing is mentioned that Plantae and Animalia, they were kept constant. Okay, and by the understanding of organisms, the kingdoms, okay, they were being changed. Okay, and that's how we got different types of classification system. Finally, we got this type of classification system, which is very important, five kingdom classification, which was given by R.H. Whitaker in 1969. What he did, actually he proposed this classification system and he made five different kingdoms. You can see the name, Kingdom Monera, Kingdom Protista, Fungi, Plantae and Animalia. What is the importance of this classification system? First of all, remember the name of this scientist, very important. Okay, and then what are the five kingdoms? Okay, and what is the criteria? On this question has been asked many times. Okay, what was the main criteria for the classification system used by R.H. Whitaker? Okay, so he used classification based on the different characters, different criteria. First criteria which he chose was cell structure. Okay, what do we mean by cell structure? Cell is a prokaryotic cell or is it a eukaryotic cell? Okay, it is having a primitive type of nucleus or it is having a well-defined nucleus. Okay, then next character they took was the body organization. Okay, body organization. What is the meaning of the term body organization? Okay, so if you look at the body of that organism, whether it is a single cell unicellular or it is multicellular, okay, or it is having tissue grade of body, okay, or is it having organ or is it having organ system? Okay, so what is all these things? I will tell you. Okay, then next is the mode of nutrition. Okay, so mode of nutrition, whether that particular organism kept under the kingdom, it is autotrophic, okay, or it is heterotrophic. Okay, there are certain types of autotrophic and heterotrophic nutrition that we will learn when we will learn about different kingdoms. Okay, next criteria which he chose was reproduction. Okay, reproduction, okay, how they will reproduce, and phylogenetic relationship, okay, how they are related evolutionarily. And along with this you have to remember he also used a character cell wall and its composition. Okay, from where I am telling you this, if you will check the you can say this table in the text, then you will get cell wall as a second point. So always remember and never forget about this. These are the you can say main criteria for classification which you have to remember. Question can be asked from this, that what were the main criteria of classification or for classification used by R.H. Whitaker? They will give you options like cell structure, body organization, mode of nutrition, reproduction, phylogenetic relationship, cell wall composition. Okay, then they can give some more options like you can say nature of genetic material. Okay, or you can say presence of pigments. Okay, anything they can ask and you have to mark that how many options are correct. Okay, let's move on to this table which is very important. You have to understand it, very important for you. Okay, see, here you can see the five kingdoms are mentioned, and here you can see over here that characters are mentioned. So first kingdom you can see Kingdom Monera. What all things you are going to read in Kingdom Monera? Okay, in Kingdom Monera, always remember you will read about bacteria. Okay, bacteria. Okay, bacteria which include your eubacteria, okay, eubacteria and archaebacteria. Okay, eubacteria and archaebacteria. In eubacteria, you will learn about blue-green algae. You will learn about mycoplasma. Okay, you will learn about the heterotrophic. Okay, heterotrophic bacteria. Okay, you will learn about the You can say yes, yes, and actinomycetes also, they come into this group. Okay, so these are the organisms which I am talking about when I am saying about bacteria. And in archaebacteria, you will learn about the halophiles which grow in salty conditions. Okay, then you have your methanogens, and you have your thermoacidophiles. Okay, thermoacidophiles. Okay, so these are the groups, these are the different organisms which I am talking about if I am talking about Monera. Okay, so what is the major character of this member of this group? Okay, so in kingdom Monera, cell type, okay, cell type will be prokaryotic. Only one kingdom is prokaryotic. Primitive type of nucleus, all the members they will have. Other all four kingdoms are eukaryotic, right? So if I say that this is a prokaryotic type of cell, it means I have a cell. Okay, it's a rough figure, so you can see it's having a cell, and in this cell it is having a genetic material. Okay, this genetic material is present, which is mainly double stranded circular single circular DNA. Okay, and let's suppose this is a bacterial cell. Okay, this is a bacterial cell, right? So you can see here this condition is a prokaryotic condition. Okay, prokaryotic cell it is. Okay, on contrary to this, if I want to make a distinction with eukaryotic cell, so when I'm making a simple eukaryotic cell, I will make a cell. Okay, I will make its genetic material, and then I will package this genetic material inside a nucleus. I will package this genetic material inside a nucleus, right? This is a nucleus. Okay, and this is the double stranded DNA. Okay, so this is a eukaryotic condition. Okay, this is a eukaryotic cell, and this is a, you can say, true type of nucleus. Here there is no nucleus in this condition. Can you see any nucleus? No, the genetic material is lying naked. Okay, naked in the cytoplasm. Okay, and here the genetic material is enclosed inside a nucleus. Okay, it is not naked, it is enclosed by a nuclear envelope. Okay, this is the nuclear envelope. Okay, a part of nucleus. Now in kingdom Monera, you have read that the cell type is actually prokaryotic. Okay, cell type is prokaryotic. Okay, now in other groups, you can see in kingdom Protista, it is eukaryotic. In fungi, eukaryotic. Plantae, eukaryotic. Animalia, eukaryotic. So in the Protista to Animalia, you will find this condition. In others, you will find this condition. Okay, other means only kingdom Monera. Okay, only kingdom Monera. Now next important feature, cell wall. If you talk about cell wall, okay, so cell wall will be non-cellulosic, means there will be no cellulose here. Okay, no cellulose will be present in the cell wall of kingdom Monera. Polysaccharides and amino acids will be present. Okay, okay, so here I am writing that in eubacteria, okay, in eubacteria members, actually the cell wall is made up of your peptidoglycan. Okay, cell wall is made up of peptidoglycan. Okay, what is this peptidoglycan? This peptidoglycan is you can say polysaccharide. Okay, it is a polysaccharide, peptidoglycan. Okay, and in this you can see we have amino acids. Peptido means some peptide will be present, and peptide will be made up of amino acids. And then we have glycan, which is nothing but a polysaccharide. Okay, so you have understood what is the meaning of this polysaccharide plus amino acids. Okay, and in certain members like archaebacteria, it is made up of pseudomurine. Okay, pseudomurine. Okay, so this you have to remember that in archaebacteria, cell wall is made up of pseudomurine. In eubacteria, it is made up of peptidoglycan. And in mycoplasma, which is a member of prokaryotes, kingdom Monera, right? In mycoplasma, cell wall is not present. So no cell wall. It is a wall-less organism, no cell wall. Very important question has been asked many times. Which is having no cell wall, in which is a prokaryotic cell having no cell wall? Okay, so mycoplasma. Okay, so this is about kingdom Monera. Now let's come to kingdom Protista. In kingdom Protista, cell wall is present in some. Okay, in kingdom Protista, what are some members in which cell wall is present? So, some members in which cell wall is present, always remember, in chrysophytes cell wall is present. Okay, silica depositions we will learn. Okay, in dinoflagellates, cell wall is present. In these two groups, cell wall is present. Others, it is absent. Okay, now in kingdom fungi, okay, cell wall is present and it is made up of chitin. Okay, in plantae, cell wall is present, it is made up of cellulose. And in animalia, cell wall is absent, no cell wall. Okay, then we have nuclear membrane. If you talk about nuclear membrane, so obviously if a cell is a prokaryotic cell, can you see any nuclear membrane here? Can you see any nucleus here? No, right? If nucleus is not present, then nuclear membrane will also be not present. Okay, what is this? This is actually nuclear membrane, or I can write nuclear envelope, right? So, in prokaryotes, okay, like Monera, nuclear membrane will be absent. In all others, it will be present. You can see here, okay, in your Protista, in fungi, in plants, in animals, you will get this condition where nuclear membrane is present. So, this character you have understood. Then let's come to body organization. If you talk about the body organization, Monera, which are actually unicellular, okay, unicellular prokaryotes. So, you will see that here body organization is cellular. Okay, some colonial forms are also present, but then also they will be individual cells which will be living together. So, body organization cellular. Okay, in Protista, they are called as unicellular eukaryotes. Okay, this one is unicellular prokaryote in Monera, and here unicellular eukaryotes, okay, in Protista. Okay, so here also you will get organisms which are made up of single cell. Okay, now let's come to the multicellular kingdom, that is fungi. Here it is multicellular. Okay, multicellular you can see. Okay, and if you talk about the cellular you can say organization, so cells are organized into loose tissues. If you have touched a fungi or any other type of mushroom, let's suppose, okay, any type of fungus you have touched mushroom, then you will feel that it is very soft, spongy type of you can say structure, right? So because there the tissues are loose, they are not compactly arranged. Like if you touch wood, okay, then what you will feel, that wood is very compact, tough, okay, because the cellular arrangement there, it is very tight, packed. Okay, here it is loose. Okay, in case of fungi. Now here you can see in case of plantae, we have body organization, tissue and organ. So in plants you get tissues. You have read about xylem, phloem, right? They are all tissues. Okay, you have learned about photosynthetic tissue, parenchyma, chlorenchyma, so many things you have read. Okay, so these are the tissues which you can see in plants. And organs, what are the organs? Leaves, roots, stems, okay, flowers, fruits, these are all the organs which you can see in plant. But have you seen any organ system? No, right? Organ system you will see in animalia. Okay, there you will see tissue, organ and organ system, all the three levels, higher levels, okay, you will find in animals. So this you have to remember. Question will be asked, organ system level of body organization is found in, or exclusively found in, okay, only found in, what is the answer? Animalia. Okay, right. Now the last and very important character, mode of nutrition. Let's finish off this mode of nutrition. Here you can see in Monera so many different modes of nutrition are present. So this kingdom is called as metabolically, okay, metabolically most diverse kingdom. Okay, metabolically this is the most diverse kingdom. Why? Because here you can see different types of metabolism. See, members, they can be autotrophic. Okay, autotrophic, who will be autotrophic? So blue-green algae or cyanobacteria, they will be autotrophic because they are having chlorophyll A, right? Chemosynthetic as well as photosynthetic. So autotrophic mode of nutrition is also of two types. Okay, photosynthetic, where photosynthesis will happen. Okay, so in blue green algae actually photosynthesis will happen, and chemosynthesis, where any inorganic substance will be used, it will be oxidized to form ATP. So all the nitrogen fixing bacteria, they will be having chemosynthetic, okay, chemosynthetic type of autotrophic nutrition. Okay, example, any nitrogen fixing bacteria. Okay, heterotrophic, okay, where they have to get dependent on some other food source. So here you can see it can be saprophytic or parasitic. Okay, saprophytic or parasitic. So they can, you can say they will convert the complex organic material into inorganic material, and then they will, you can say, use it for their nourishment. Saprophytic mode of nutrition. And parasitic, obviously they will live in a host and they will derive nourishment from that. Okay, so when we will read about kingdom Monera in detail, we will cover all these points, but this you have to remember, metabolically most diverse kingdom it is, because it is having all the different possibilities of nutrition which are shown by this kingdom. Okay, then kingdom Protista, you can see it is mostly autotrophic and also heterotrophic. Okay, some members are autotrophic like dinoflagellates, chrysophytes and euglenoids. These three groups are photosynthetic. Okay, here photosynthesis is possible because pigments are present, right? Because they have chlorophyll A. Along with that, we have some other pigments. Okay, and some are heterotrophic. Heterotrophics like your slime molds and your protozoans, these are heterotrophic groups which are present in kingdom Protista. Then you talk about fungi. So obviously, no doubt, it is the heterotrophic mode of nutrition which they will show, because they are devoid of any chlorophyll. They can't make their food, so it is heterotrophic, right? And along with heterotrophic, you have to remember, see, I am marking this again for you, and it is heterotrophic here in kingdom fungi, and both saprophytic and parasitic type of heterotrophic nutrition you will see. Plantae, you can see in plantae we have autotrophic mode of nourishment. Okay, nutrition. Autotrophic, and all are photosynthetic except a few. Except a few. What are the exceptions? Exceptions I am writing. You can see Cascata. Okay, what is Cascata? Cascata is a parasitic plant. Okay, parasitic plant. Why? Because it is having no chlorophyll. So let's suppose this is you can say some branch of a tree, and on this branch this cascata will, it is a vine, right? So it will grow its hostoria, and it will drive the nourishment from this branch, and it will take the food and water from this branch. And this is a parasitic type of interaction which it shows because it is having no chlorophyll. Along with cascata, there are certain, you can say, certain plants. Okay, certain plants are there like your insectivorous plants. Okay, insectivorous plants. Okay, insectivorous plants like Nepenthes, Venus flytrap, they take nitrogen, okay, from insects. Okay, they take nitrogen from insects. That is why they are called as, you can say, semi or partially. Okay, better word is partially. Okay, they are partial heterotrophs. Okay, partial heterotrophs. Okay, heterotrophs. So yes, you have to remember that these insectivorous plants, they will do photosynthesis. They have the leaf-like structures. They will do photosynthesis. They have pigments, but they are deriving some other important, you can say, nutrients from other sources, right? That is why they are semi-heterotrophs. Okay, so majorly, all in all, kingdom Plantae is autotrophic, but few exceptions are there. Cascata, parasitic plant, it is not autotrophic. It is heterotrophic, showing parasitic mode of nutrition. And insectivorous plants, they are partially heterotrophic. They will prepare their glucose by photosynthesis, but some other things they will do. They want nitrogen. They need nitrogen, so they will get the nitrogen from the, you can say, insects, right? Now Animalia is strictly, it is exclusively heterotrophic group. Okay, in particle form, animals, they can get the food, they can take in the food, holozoic type of nutrition, and saprophytic nutrition is also observed, right? So here you can see, okay, you can see very well we have discussed some important characters of 5 kingdom classification, on basis of which characters these different organisms were kept in these 5 kingdoms. Okay, so now if I ask you a small question, okay, a small question, that which is that classification, which is that kingdom, okay, kingdom which is exclusively heterotrophic, which kingdom is exclusively heterotrophic? What should be your answer? You can see, pause the video and tell me. Okay, so exclusively heterotrophic means only heterotrophic, and here is your answer. Okay, in only these two kingdoms you can see heterotrophic nutrition, right? So kingdom fungi and kingdom animalia is the answer. Okay, now another question, kingdom which are exclusively autotrophic, no other type of nutrition, autotrophic. Yes, kingdom plantae. Okay, exceptions you have to keep in your mind, but if you go with the major members which are you can say having this mode of nutrition, so autotrophic mode of nutrition is shown in kingdom plantae. So questions will be asked like this, right? Okay, now we have to talk about 3-domain system. Here you can see, after the success of 5 kingdom classification system, there was a scientist, okay, Carl Woos. Okay, what he did? Carl Woos, he divided kingdom Monera. Okay, he divided kingdom Monera. Okay, into 2 groups. Okay, what are the 2 groups? 2 groups are Eubacteria and Archaebacteria. Okay, Eubacteria and Archaebacteria. Okay, he divided all the different members of the Monera into these 2 groups. So what happened actually, that there was establishment of 3 domains of life. 3 domains of life. What are the 3 domains of life by Carl Woos? So 3 domains of life were eukarya, archaea, okay, archaea, and you can say it was eubacteria, or you can say bacteria. Okay, in bacteria all the true bacteria were kept. Okay, all the true bacteria were kept. Okay, you can write it as bacteria. Okay, and in archaea all the archaea were present, but in eukarya, starting from the protista, okay, starting from the protista, or protista, fungi, plants and animals. Okay, protista, fungi, plants and all the animals were kept. So what you can see here, here all the archaebacteria were present, and here all the true bacteria were present. So you can see these are 3 different domains of life. Actually eukarya should be kept here, and eubacteria and archaebacteria should come here. Okay, according to phylogeny, but roughly I am telling you that 3 domains were present, the archaea, bacteria and eukarya. After that you can see 6 kingdoms also. What are the 6 kingdoms which you can see? See, protista, okay, fungi, plants, animals, archaebacteria and true bacteria. So if you go with phylogeny, so it should start with prokaryotes, but here I'm just giving you a brief introduction that how they have divided it. So you can see here that 3-domain system has also been proposed, that divides the kingdom Monera into 2 domains, leaving the remaining eukaryotic kingdoms in the 3rd domain, and thereby a 6-kingdom classification, right? So 3-domain system or 6-kingdom classification. What you have to remember, that 3-domain system and 6-kingdom classification are same, proposed by Carl Woos. He divided Monera into 2 groups, and you can see all the remaining 4 were kept under a category eukarya, and the other 2 archaebacteria and eubacteria were kept into 2 different domains, right? So that is how we can say it is a 3-domain system or a 6-kingdom classification. Okay, now we can solve some questions. First question you can see, in Whittaker system of classification, prokaryotes are placed in kingdom. If you go by R.H. Whittaker's 5 kingdom classification system, okay, where you will put the prokaryotes? What are prokaryotes? Which are having no well defined nucleus, right? Okay, so all the prokaryotes were kept under kingdom Monera, right? Kingdom Protista, all the unicellular eukaryotes. According to R.H. Whittaker, I am telling, okay, according to 5 kingdom classification. Okay, do not forget, right? Not the early classifications, 5 kingdom classification, right? Unicellular eukaryotes. Monera, unicellular prokaryotes. Remember, I have told you. Plantae, multicellular eukaryotes. Okay. Autotrophic. Okay. So many things we can write. Multicellular eukaryotes. Okay. Heterotrophic. Okay, so we got the answer. Now, according to 2 kingdom classification, Saccharomyces should be kept under which kingdom? Okay, 2 kingdom classification proposed by Linnaeus. Okay, according to this classification system, where should I put Saccharomyces? What is Saccharomyces? It is yeast. What is yeast? It is a eukaryote. Okay, which eukaryote? It is a unicellular fungi. Okay, and fungi were kept under fungi, they were having cell wall. Okay, fungi, they were having cell wall, so that's why they were kept under kingdom Plantae. So where I should keep Saccharomyces yeast? According to 2 kingdom classification system, I should keep it in fungi. No, right? Fungi was not a kingdom there. Only 2 kingdoms were there, Plantae and Animalia. So we have to keep this under kingdom Plantae. Pick up the wrong statement. Protists have photosynthetic and heterotrophic modes of nutrition. Some fungi are edible. Nuclear membrane is present in Monera and cell wall is absent in Animalia. Okay, so with that table, you can solve this question. Okay, protists, they were photosynthetic and heterotrophic both, right? We have learned that chrysophytes, dinoflagellates, euglenoids, they were photosynthetic. Slime molds and what else? Protozoans, they were heterotrophic. Right statement. Some fungi are edible. Yes, you eat mushrooms, right? You eat mushrooms. This is correct. Cell wall is absent in Animalia. Animalia, animals, do they have any cell wall? No, right? This is a correct statement. Nuclear membrane is present in Monera. Monera is a prokaryotic cell. What is Monera? Prokaryotic cell. And prokaryotic cells, they lack any nuclear membrane. Okay, there is no nuclear membrane. Okay, this is a prokaryotic cell, and in this prokaryotic cell, here you have the genetic material present. So where is the nuclear membrane? No nuclear membrane. Okay, this is the correct answer. Cell wall composition of fungi. Okay, what is present in the cell wall of the fungi? In the cell wall of fungi, we have chitin. Okay, a complex polysaccharide. Okay, peptidoglycan is not present, murin is not present, cellulose is not present. Okay, chitin is present. Right. So this is the correct answer. So this was the discussion on the first part, that was the introduction part on the chapter biological classification. I hope you have understood. We will come again with continuing this chapter. Thank you very much. Solve the questions and learn with me. Thank you.